no, we don't. But I got your attention! The natives are restless here today, it sounds like. So, how's everybody doing? Are you happy you don't have an exam on Wednesday? Yeah. Me too. So, I finished up prostaglandins last time, and we did go through prostaglandins at not much depth, I'll be honest with that, partly because there is a lot of complexity to it. If we were teaching this class in medical school, we'd probably spend a whole week on prostaglandin just because of the various things that they do and how they do them. So I know they're interesting compounds. They're ugly-looking compounds, but that's what they are. I was going to finish the day the other day with a song, so I thought we might start the day today with a song about prostaglandins. That'll get us off to it, get some of that energy off here. This is a song about prostaglandins that I wrote to the tune of the Broadway musical Oklahoma. And you guys know Oklahoma where the wind comes sweeping down. Okay, so I want to hear you. You guys have been good about this lately. Let's sing about prostaglandins. Prostaglandins, the icosanoids creating pain, are the ones to blame when you get inflamed and ouch, they hurt inside your brain. Prostaglandins, every throb and ache gets magnified. If you hope to win, cyclooxygen's generation's got to be denied. The Biox has all been recalled, so go get yourself Tylenol, and if you blame PG aches and days, we must complain that you make the aches prostaglandins. Prostaglandin, D2F1, G2E2, prostaglandin, it's you. Okay. Thank you. I put my heart into that song. I want you to know that. So, okay. So, we are now embarking on the last topic of metabolism that we shall work on. So, if you wanted to clap about something, that might be it. You guys will clap for anything today, won't you? Good mood today, aren't we? We should have a happy hour or something. Wouldn't that be cool? You know, if we could find a place that would be big enough, it would really be fun to go do a happy hour. You know, wouldn't it? Woodstocks? Maybe at the end of the term. I mean, we've done 2 terms together. We could do like a happy hour at Woodstocks. Would that be cool? Okay, that's fine with me. I'll drink to that. We'll do that. We'll schedule something at Woodstocks. Okay. I never should have said that because now the energy level is going to go back up again, right? So, there's beer. No. Okay. You can see I don't want to do this any more than you guys do. All right. Let's go. Yeah. I'm sure you would have no shortage of applause or votes for that. So, okay. We're getting ready to talk about nucleotide metabolism. And nucleotide metabolism brings together a lot of things that you've seen in other metabolic pathways. And it brings things together, I think, in a very interesting way. Okay? So you've seen how cells spend a lot of energy on controlling energy pathways, for example. So they invest ATP, they have all kinds of structures that respond to allosteric factors, that respond to covalent modification, that do some very interesting things so that we have the proper amount of energy inside each of our cells. And nucleotide metabolism is interesting because, first of all, nucleotides are, in fact, energy carriers. ATP, GTP, we've seen these things being important for energy production. We know, of course, that the nucleotides are also very important for the synthesis of RNA and DNA. And it's towards that end that the cells are what I would describe as control freaks for the levels of each of these nucleotides. What we're going to see in nucleotide metabolism is cells control the levels of nucleotides more carefully than anything else they can control in the cell. All right? They've got elaborate controls, and I don't mean intricate. They're not as complicated of controls, for example, as we saw in gluconeogenesis and glycolysis and glycogen metabolism. They're not that complicated, okay? But they're very elaborate so that the cell does not get any nucleotide in too great or too small of a quantity. Okay? That's a very important thing, because we know today that when cells are disrupted in how much or how many of any given nucleotide they have too much or too little of one, that cell is very prone to mutation. Okay? So the reason that cells have these controls is largely to avoid mutation. Right? So that's a very important consideration for nucleotide metabolism. Nucleotide metabolism occurs at two levels. One level is what we call de novo synthesis. And de novo synthesis simply means starting from scratch. Literally, de novo means from new. All right? That means we start with very simple compounds, and as you're going to see, we can make nucleotides from incredibly simple things, like carbon dioxide, like amino acids. Okay? And it's not surprising that we can imagine in a primordial Earth those simple compounds being present and coming together chemically, as we'll see they happen in our case in modern day with enzymes, but how they could come together chemically and make, in fact, make the nucleotides that we see. There are indications, for example, that simple nucleotides actually are in meteorites that fall from space. Okay? So, these reactions that make these things are abundant, they're present everywhere, and we can see them happening. The second type of nucleotide synthesis that we talked about, the first being de novo, the second one we call salvage. And salvage is, as its name suggests, an ability to use pieces of nucleotides to rebuild. Now that makes a lot of sense from an energy perspective. Instead of taking everything from scratch and starting up, what if in the digestive process, okay, I eat some food and I get a bunch of guanine base present? Wouldn't it be nice to use that guanine base to make guanine nucleotides? Okay? So salvage makes a lot of sense from an energy perspective, and cells go through a lot of salvage pathways. We're going to cover them in a little bit of depth, not a lot of depth. But the salvage pathways turn out to have some very important and interesting human health implications as well when they're defective. So, the scheme on the board just shows you those two pathways, salvage and de novo. We're not going to worry about the details that are up there at the moment. Okay, I know that most of you have had this in basic biology, but I find that when I ask these questions on the exam, I still get a pretty decent number of people who don't get them right. So I want to make sure we go over and understand the nomenclature that we have with respect to nucleotides, nucleosides, and bases. Okay? So, there are three names I've just given you right there. Nucleotides, nucleosides, and bases that I would expect that you would be able to tell me or even roughly sketch out. I'm not going to ask you to go draw the exact carbons and the exact nitrogens and so forth, but I think you should be able to at least sketch that for me. Okay? So let's start at the simplest level. The simplest level is a base. It's a base that people frequently confuse with a nucleotide. A nucleotide contains bases. Adenine, guanine, cytosine, thymine, and uracil are all bases. Okay? When they're contained in nucleotides, they can form base pairs with the corresponding other bases in other nucleotides, and that's how we get double helices. Okay? But a base is just that. It has nothing more than that. It's just the base itself. If I take that base and I attach it to a 5-carbon sugar, that 5-carbon sugar would either be ribose or deoxyribose. If I attach that base to a 5-carbon sugar, I make what we describe as a nucleoside. Okay? A nucleoside, and you notice that the name changes. The name changes from being a base, adenine, to being an adenosine, as we see here. So So if I talk about adenosine, I'm talking about a nucleoside. If I put the base onto a deoxyribose, then I make, of course, the deoxynucleosides. And these would be deoxyadenosine, deoxyguanosine. You'll notice thymidine has its own sort of separate designation. And you may call that either thymidine, or if you want to call it deoxythymidine, I won't count that incorrect. That way you just memorize one basic thing, deoxythymidine. People say it's redundant because thymidine you don't have a ribo version of. Okay? To which I say, well, yeah, we do. In some cases, have a ribo version of thymidine. Turns out that some tRNAs have a ribo version of thymidine. In any event, if we take now that nucleoside and we put at least one phosphate onto it, we make a nucleotide. Okay? If that happens to be a compound that contains a ribose sugar, of course it's a ribonucleotide, and ribonucleotides are used for making RNA. If it contains a deoxyribose, it makes a deoxyribonucleotide, which is used for making DNA. Okay? Now, one of the places where students trip off is they say, well, what's a nucleoside monophosphate? And they'll hear nucleoside and they'll say, it's a nucleoside. Right? But a nucleoside monophosphate means it's a nucleoside with a phosphate on it. That means a nucleoside with a phosphate on it is a nucleotide. Right? So, if I say I have deoxyadenosine monophosphate, I'm talking about a nucleotide. Once it has a phosphate, at least one phosphate, it becomes a nucleotide. Make sense? Mm-hmm. Okay. So make sure you have those general things down. I'm not going to ask you to draw exact structures, as I said. The sugars that we find in... Well, that was good. Did they pull that on me? Oh, those turkeys. Okay. No, I mean, this is from my own textbook. I wrote a textbook. I was a co-author on a textbook years ago, and I was using figures from my textbook, and they've yanked them. So you're going to have to trust me on this. Okay, we talked about ribose last term. I'm not going to make you draw the structure, but you should know that ribose is a pentose, meaning it has 5 carbons. It's a very simple structure. When you memorized it last year, I'm sure you remembered the fact that it had 5 carbons, oxygen at the top, 2 hydroxyls hanging down. That was ribose. Okay? Deoxyribose. Essentially the same as ribose, with the difference that the carbon number 2, instead of having a hydroxyl group, has a hydrogen. That's the deoxy part. It's lacking an oxygen at position 2. Okay? Now, it turns out we don't typically have deoxyribose free in the cell. It's not something that the cell synthesizes as such. Instead, deoxyribose is made on a nucleotide. Okay? And we'll see how that happens later in the lecture about that. So deoxyribose is not made separately, it's made as part of a nucleotide. So I am guessing now that all of my bases and all of my cute little figures are all those turkeys. I just did that just recently. That's okay. All right. Well, I will get some more figures posted for you. I apologize that those are not there, but
Kevin, are there any the PowerPoints that you can get to on the website?
If I go into PowerPoint, it actually confuses things up here, so I prefer not to do that. That's fine. I'll get you guys some figures that you can see those. Suffice it to say, if we think about the structure of a nucleotide, we can think of, here's ribose, okay, at the core. We have 2 things hanging off of it. Off of carbon number 1, that's where the base is. Okay? And off of carbon number 5, that's where the phosphate or phosphates are located. Okay? So that's the general structure of a nucleotide. A nucleoside would lack the phosphates, obviously. And the bases, since I can't even show you those, the bases I will describe to you as in the depth that I would expect you to be able to draw them. Okay? The bases are in 2 groups. They're known as pyrimidines or purines. The pyrimidines include cytosine, thymine, and uracil. Each of those has a 6-membered ring. There's some nitrogens. We're not going to worry about the exact structures, but they each have a 6-membered ring. The other category are the purines. The purines have a 6-membered ring joined to a 5-membered ring, so they're bigger. Purines include, of course, adenine and guanine. Okay. Well, that should get us at least through our stuff there. The deoxyribonucleotides have exactly the same structure as the ribonucleotides, the difference being, of course, the deoxyribose sugar instead of the ribose sugar. Okay, well, let's get on to talking about the synthesis of nucleotides. So, I'm going to start first and talk about the synthesis of pyrimidines. Pyrimidines are the simpler of the two nucleotides because they only have that 6-membered ring. I shouldn't say 6-carbon, but 6-membered ring. Okay? And as you can see here, they come from fairly simple compounds. This is a schematic. You see, for example, that carbamoyl phosphate, which contains this nitrogen and this carbon, is made very simply from bicarbonate and ammonia. Two very, very simple compounds, two very abundant compounds. That takes some energy to put those guys together. But when you put those guys together, you've got carbamoyl phosphate, and I'll show you how that goes in a minute. If I combine that with aspartic acid, I've basically made that 6-member pyrimidine ring. Okay? So, very simple compounds that are there. Right? That pyrimidine ring is combined with a modified ribose sugar called PRPP. PRPP stands for phosphoribosyl pyrophosphate. Big name. Okay? Phosphoribosyl pyrophosphate. I think I do have a structure I can show you of that. If I don't, then I'll have to describe it as well, but I think I've got that on here. At that point, we have joined a sugar, a phosphate, and a base. At this point, we have a nucleotide. Now, it turns out that nucleotide that we get first is actually a nucleotide of uracil. It's called UMP, uridine monophosphate. Okay? Uridine monophosphate. That's the very first nucleotide that we get. And then it's from UTP that we get the other pyrimidine nucleotides. Okay? So I'm going to show you the details about how we make that in a second, but I wanted you to have a basic understanding about how we go about doing that process. Okay. All right, so here is the synthesis of that that carbamoyl phosphate. And you guys have actually seen some of this stuff, at least not this stuff before, but the things that this is used for before. And you'll see that coming up in just a second. So don't sweat too much what's on this figure. I want you to understand, first of all, what the enzyme is doing. So we're ultimately going to make carbamoyl phosphate. Again, we're not sweating structures here, so don't sweat the structures, but we're interested in the compound being carbamoyl phosphate. All right? To make this compound, okay, we have to get an ammonia, and that ammonia comes from the amino acid glutamine. Okay? In the process of making carbamoyl phosphate, we make carbamic acid. And carbamic acid turns out to be very unstable in water. Extraordinarily unstable in water. Now, last term, you may recall when I talked about the nucleoside monophosphokineases, we talked about those guys having a lid. Remember the lid that came down and closed when the substrate's bound? The reason that lid closed was that phosphate could just freely go out into solution, but the lid kept it from going out into solution and constrained it there. So we saw an example there of a protein structure that basically ensured we got the compound that we wanted. Remember that? We also talked last term about triphosphate isomerase. Triphosphate isomerase was an enzyme in the glycolysis pathway that we described as a perfect enzyme. And it was a perfect enzyme because its speed, the speed with which it did its reaction, kept an unstable intermediate from being around for very long. Okay? Now we see in this reaction, and we see in this enzyme, a third type of structural consideration for an enzyme to ensure that the cell gets the product. And this is an interesting one. This one uses a tunnel. Okay? The materials go in one end, up here. The reactions are catalyzed in a couple of places on the inside. And the beauty here is on the inside, water is not present where the unstable molecule can get broken down. The final product, carbamyl phosphate, is spit out at the other end, and it's not unstable in water. Okay? So this enzyme is very, very useful for protecting and making sure that we get the product that we're after. That enzyme is carbamyl phosphate synthetase. Okay? Where did I put it? There it is. Okay, carbamyl phosphate synthetase. It uses what your book describes as channeling. Other people call it tunneling. They're the same phenomena. In each case, this molecule is being made as it's going through the enzyme. Okay. Now, one of the things that your book sort of skips over here, which I find a little surprising, is the fact it doesn't show the next reaction, which actually makes this guy up here. Okay, so at least I think. Let me make sure before I blather that out there. I'm pretty sure we don't have that in. Okay, I'm sorry. We do have that there. Okay, so in the next reaction, we take that carbamyl phosphate and we combine it with aspartate. So we're on our way to making this six-membered ring. Okay? Yes, sir.
What would carbamic acid go to in the presence of water?
The phosphate would come off. Yeah. Okay. And I can't remember what that would be, but the phosphate is the unstable part of that. Okay. So what we take here is we add this carbamyl phosphate and we combine it with aspartic acid to make now what's beginning to look like that six-membered ring. This reaction you've seen before. Last term we talked about the enzyme aspartate transcarbamylase, or you learned it as ATCase. So ATCase catalyzes the formation of carbamyl aspartate. Okay. Now, you may remember from our discussion of the ATCase last term that ATCase was an enzyme that was an example of allosteric regulation. It was turned off by the end product of the pathway, and the end product of the pathway turns out to be CTP, as we shall see. So, aspartate transcarbamylase, ATCase, is inhibited by CTP. The enzyme is allosterically activated by the corresponding, or not by the corresponding, but by the purine ATP. Now, at the time last term when I talked about it, I probably did not mention the fact that this is the first example that you see of what I describe as balancing in the cell. The cell is balancing the relative amounts of purine and pyrimidines. This enzyme is controlling that. If pyrimidines get too high, the enzyme gets turned off, and the cell quits making pyrimidines. On the other hand, if purines get too high, then it activates this enzyme and stimulates the production of pyrimidines. And of course, when we make DNA, we have to have pyrimidines and purines. So this is the first example we see of balancing the relative amounts of nucleotides in the cell. As I said, ATCase balances the relative amounts of purines and pyrimidines. By the way, the pathways that I'm showing you here, these de novo pathways, that I'm showing you are for making ribonucleotides. We'll see later that deoxyribonucleotides are made from the ribonucleotides. Okay. Well, there's a couple of reactions. We're not going to sweat the details of the reactions. This is not like glycolysis. We're going to go and do that. We see loss of a water. We get a closed ring structure called dihydroorotate. And we get reduction of that dihydroorotate to make, we get oxidation of that dihydroorotate to make orotate. Blah, blah. Now, This reaction adds that phosphoribosyl pyrophosphate, that PRPP molecule I talked about. There it is. You can see the central part of it here is the ribose. There's the ribose structure. We see the pyrophosphate hanging off of carbon number 1. And we see the other phosphate hanging off of carbon number 3. Okay? Notice that in the joining of this orotate with the PRPP, that this alpha configuration is being converted into a beta. Okay? So when we draw structures of nucleotides, we will have the base above the ring of the ribose or deoxyribose. That is, this anomeric carbon is always in the beta configuration. Okay. And next, we do something very interesting. We take orotadylate, which technically is a nucleotide. You don't see it in any of the nucleic acids. Again, I'm not worried about the name here. But we're making uridylate, which is the same thing as UMP. Okay? I think if I were you, I would remember UMP. Uridylate is kind of an awkward name. Okay? Technically, it's uridylic acid. That's not what we're going to work with. So UMP is the way you know it. This is uridine monophosphate. There's the uridine. Well, there's the uracil. There's the ribose, and there's the phosphate. That's UMP. Okay. So at this point, we have a nucleotide that's going to appear in RNA. So for the first time, we have that. Now, this reaction I show you because it's interesting, and it's interesting because of the enzyme that catalyzes this reaction. This enzyme catalyzes the decarboxylation of this carbon right here off of the orotate that's over here. This enzyme is orotate decarboxylase, orotadylate decarboxylase, but again, the important thing isn't so much the exact name of that guy as much as it is what it's doing and how fast it's doing. This enzyme was one I talked about last term when I talked about enzymes, and I said how enzymes can speed up a reaction up to 10 to the 17th fold. This is the enzyme. This enzyme catalyzes a reaction that goes from once in 78 million years to about once a second. Okay? This enzyme is incredible at speeding up this reaction. Okay? So it's a phenomenal enzyme. Okay. Now, at this point we have UMP. Okay? So, of course, cells don't use the monophosphate to make RNA and DNA. They have to have triphosphates. We'll talk about DNA replication and RNA replication later. But the starting materials are not monophosphates, they're triphosphates. So we have to get triphosphates. To go from monophosphates to diphosphates, we have to use our friends, the nucleoside monophosphate kinases. These were the guys with the lids. Okay? And so a nucleoside monophosphate kinase will be specific for the monophosphate. So there'll be one for UMP, there'll be one for CMP, there'll be one for GMP, and there'll be one for UMP. Okay? I didn't talk about T because T is a deoxy and we don't start those here. Okay? So UMP, GMP, CMP, AMP. All of those guys will have their own individual nucleoside monophosphate kinase. They're all going to use very similar mechanism. And notice they're going to use a triphosphate energy source, ATP. Okay? So it means we've got to have more ATP than any of the other triphosphates present in the cell. Well, you guys know that happens because you get that from oxidative phosphorylation. Oxidative phosphorylation is not producing GTP. It's not producing CTP. It's producing ATP. So we have plenty of ATP to do this. When we do this, we start out with a monophosphate and a triphosphate. We end up with a diphosphate and a diphosphate. Okay? Well, a diphosphate is getting there, but it's not quite as far as we need to get. We need to convert that diphosphate to a triphosphate. Now, notice I've called this XDP plus YTP. Gives XTP plus, that should be a YDP. I don't know why I've got that. That should be a D right here. I'm sorry. That's my mistake. I can't blame the book this time. Okay. That it should be a YDP. So we've basically moved the phosphate from the Y nucleotide to the X nucleotide. Now, why did I call it X and Y? The reason I call it X and Y is that this enzyme here, nucleoside diphosphate kinase, you also hear it called diphosphokinase, you're more likely to call it NDPK. NDPK. Now, NDPK catalyzes this reaction for every diphosphate and every triphosphate. So X could be a G, and this could be a C, for example. X could be a U, and Y could be a T. It doesn't matter. So any nucleoside diphosphate can be converted into a triphosphate using triphosphate energy. So everything that takes every single diphosphate to triphosphate is converted by this enzyme. Okay? That's an important consideration because that also provides some balance. If I have too much triphosphate of one kind and not enough diphosphate of another, now I have a way of balancing how much of the individual triphosphates that I make. Okay? So I don't have figures to show you these, partly because your book doesn't have that, but you can, I think, get the idea about what's going on with those. Yes, Stephanie?
So SBP is the phosphate from YTP.
Yes, it is.
So in the other side of the reaction.
That should be a D right there.
Okay.
Yeah. Other questions? Okay. Well, we've gotten ourselves, so if we were following, for example, UMP, we've just taken ourselves from UMP to UDP, take UDP, and over here we would have UTP. Okay? So it turns out UTP is the first triphosphate that's made. It's the first triphosphate that's made. So UMP is the first nucleotide that's made. UTP is the first triphosphate that's made. Now, your cells, I shouldn't say you're not your cell, but your cells take UTP and convert it to CTP. Okay? This reaction is catalyzed by an enzyme known as CTP synthase. That's an important enzyme because this enzyme is also regulated. It's actually regulated by how much CTP there is there. It gets turned off if there's too much CTP. Okay, so this reaction converts this oxygen to an amine group up here. That's the only thing that's happening in this reaction. And it's a pretty straightforward reaction. So at this point we have two triphosphates. You'll notice that the CTP was made without going through a monophosphate or a diphosphate stage. It went directly from UTP to CTP. And we're still talking about ribonucleotides. In fact, I'm not going to talk about deoxyribonucleotides until the lecture on Wednesday. Not the Monday. Today is definitely not Monday, is it? Okay, questions about that? At this point, we have finished the de novo synthesis of the ribonucleoside triphosphate pyrimidines. Okay? All right. Now, there are some reactions of salvage that are important. I talk about salvage down later below, so I'm going to save salvage until I talk about all the medical implications for last. So I have it up here in the order that your book has it, but I'm going to talk about salvage reactions last. Instead of talking about salvage at this point, I would like to turn our attention and talk about de novo synthesis of the purine ribonucleotides. Okay? The purine ribonucleotides are a little bit more complicated. There's that 6-membered ring joined to a 5-membered ring. So we see it's a bigger, bulkier thing. And we see that the atoms within the rings come, again, from fairly straightforward things. Notice this is a very big name. I'll explain that significance in a second. But ignore the big names for the moment. You see glycine. You see carbon dioxide. You see a nitrogen from aspartic acid. You see a nitrogen from glutamine. And you see a ribose hanging down here. Okay? These big mouthful names are molecules called folates, and the only reason that they have a big name is they do have a kind of a complicated structure, but all they're doing is donating a single carbon. Okay? So a single carbon source is really all that's needed to put those two carbons in there. So again, we see very, very simple things that come together to make the bases that make the purines. Okay? The purines are made in a fundamentally different way from the pyrimidines. In the case of the pyrimidines, we made the ring first, and then we attached the ribose by using PRPP. Okay? It turns out that in making the purines, we actually build the base on the PRPP. We build the base on the PRPP. Okay? All right, and like the pyrimidine synthesis, the purine synthesis takes about 10 steps, and we're not going to go into detail on those steps. I will briefly show you the highlights of those steps in these two figures. Okay, now don't sweat the names. We're not going to. I can assure you, we are not going to memorize a single name out of there, other than probably phosphoribosylate pyrophosphate. That's the other thing about this figure that I don't like. This figure actually ignores the first reaction. It says reactions 1 to 6. This is actually the product of reaction 1. So it's actually, you're starting with reaction 2, not number 1. Okay. I'm picky, I guess. Well, let's talk about reaction 1. Let's talk about how we make phosphoribosylamine, because that turns out to be a very, very important regulatory step. Very, very important regulatory step. Okay? The enzyme that catalyzes the formation of phosphoribosylamine, by the way, the starting material is phosphoribosyl pyrophosphate. So we're starting with PRPP. We're starting with PRPP. In the reaction that's being catalyzed, an amine group is being put in place of the pyrophosphate. Okay? The amine group, the pyrophosphate on the PRPP is being replaced by an amine group. That amine group is coming from an amino acid. We don't need to worry about which one at the moment. Now, that enzyme has a name that you should know. It's a very important regulatory enzyme. It's known as PRPP amidotransferase. And no, we're not going to make up a name for that one. Okay? Because that's an important enzyme you're going to see in other places. PRPP amidotransferase. AMIDOTRANSFERASE. PRPP amidotransferase is a very interesting enzyme. Okay? PRPP amidotransferase is completely inhibited by the presence of ATP and GTP. When both of those are present, the enzyme is completely inhibited. When only one of those is present, the enzyme is partially inhibited. Okay? When only one of those is present, the enzyme is partially inhibited. I want you to keep that in mind, and I will talk later about the significance of that. It turns out this enzyme helps to balance the relative amounts of ATP and GTP. Okay? It helps to balance the relative amounts of ATP and GTP. If only one is present, the enzyme is still partly active, and you'll see later why that's the case. Okay. Well, we have a series of reactions. Here's some energy investment. Here's addition of a carbon. Here's some more energy investment, some more energy investment, some more energy investment. We see that making nucleotides costs a lot of energy. Okay? Costs a lot of energy in making nucleotides. What we see is they're not showing you the sugar part. That's why they have this P ribose down here. But what you're seeing is that the ring is slowly being built. Here's the ring. Here's the ring. There's the closure of one ring. And now the addition to start the formation of the second ring. All right? So these reactions, the rings, are being built on the ribose, ultimately the PRPP. Okay. Now, this set of reactions now shows what happens going further along to ultimately getting to this molecule over here called inocinate, or IMP. We'll call it IMP. All right? IMP turns out to be a nucleotide. And IMP, though it's not used by RNA polymerase, does show up in some transfer RNAs. We'll talk about that later. Okay? So we actually have a purine nucleotide at this point. It has a base, in this case inosine. It has a sugar, and it has a phosphate. So we have made that. Notice one of the things that's kicked off in the process is fumarate. Fumarate is useful, of course, for the citric acid cycle. So now we see some of the byproducts that are actually being kicked out and used in the citric acid cycle. Okay. Now, I will probably finish with this. All right. This figure shows how we make AMP and GMP. All right. It turns out that IMP is a branch point, and it's an interesting branch point. If we go the top route, what do we see? Well, we see, first of all, that we use some GTP to add an aspartate to this, and then we kick off a fumarate again. So we have another fumarate being kicked off, okay? And that leaves us with AMP. Okay? That leaves us with AMP. Down here, if we go this route, we see that we have an oxidation to make xanthylate. Xanthylate then uses energy of ATP to make GMP. Now, what I've just told you, you didn't realize it, but what I just told you was a very important consideration in balancing. To make AMP, I need to have GTP. And to make GMP, I have to have ATP. So the energy for making guanine nucleotides comes from ATP, and the energy for making adenine nucleotides comes from GTP. That sort of makes sense. What if I don't have any adenine nucleotides around? If I don't, I'm not going to have any ATP for energy, right? So I have to get the energy from the alternate purine nucleotide, and that's what I have in this case. So this is sort of helping to balance A and G. We'll see that it's not the major consideration. Okay? Another way that A and G are balanced, in fact, the major way that A and G are balanced is the fact that the enzyme that catalyzes this reaction right here, that we're not even going to name, you can relax, there's an enzyme name you don't have to know. The enzyme that catalyzes the upwards reaction is inhibited by AMP. The enzyme that catalyzes the downward reaction is inhibited by GMP. So that means that GMP inhibits its own synthesis, and AMP inhibits its own synthesis. Okay? If I have too much GMP, I'm going to shut this off. Presumably then, this reaction is going to go up here. Now, I want you to think back to the PRPP aminotransferase. I said originally that the enzyme is completely inhibited when GTP and ATP are both present. Of course, AMP and GMP are readily converted into the triphosphate, so once we have the monophosphate, we readily have the triphosphates. Okay? The enzyme is completely inhibited by the presence of those two nucleotides. If I have those present in high abundance, I don't want to make any more, so I completely shut down purine biosynthesis. However, if I have a lot of AMP but I don't hardly have any GMP, if I try to completely shut off purine synthesis, I'm still not going to have any GMP, right? So by letting the enzyme be partially active, this is going to turn off its own synthesis, so this top pathway is not going to go, and GMP will be produced completely down here until it gets high enough that it shuts off its own synthesis as well. Yes, sir?
Feedback inhibition.
Right here at this point, it is what we would call feedback inhibition because the end product is inhibiting here. Okay? The PRPP aminotransferase is a sort of a modified feedback inhibition because it takes both of those guys to completely shut it off. This is the first time you've seen this type of an inhibition. Okay, so does everybody understand that? I've got about 3 or 4 minutes. I'm going to summarize what I've told you in terms of balancing so far. There will be balancing that will also be involved in deoxyribonucleotides, but let me summarize the balancing here for you. ATCase helps to balance pyrimidines versus purines. ATP turns it on, CTP turns it off. CTP synthase helps to balance UTP versus CTP. Too much CTP turns it off. Okay? PRPP aminotransferase plays a role also in balancing purines versus pyrimidines. Because if I have high levels of GTP and ATP, it shuts off. And shuts off all purine synthesis and now allows pyrimidine synthesis to go. Because high ATP levels will turn on ATCase and get pyrimidine synthesis going. The enzymes involved in converting inocinate, that is IMP, into the individual nucleotides, are each inhibited by the end products. So the enzyme that leads to AMP is inhibited by AMP. The enzyme that leads to GMP is inhibited by GMP. These enzymes are balancing the relative amounts of the A versus G nucleotides. Okay? Now, these controls, as I said, are very, very important because cells have to be able to keep the proper amounts there. You let some of this get out of whack, and you're going to have problems. Okay. Any questions? I know there won't be at this point. Everybody wants to go home. It's Friday. See you guys on Monday.